Okay, good. So let's get started with the talk of Pavlo Lutsik.
Exactly, yeah.
On pipeline Olympics. Please.
Can you hear me well? Yeah, perfect. So thanks everyone who came for this later session. I'm enjoying this event enormously. I wanted to say I already met more friends than I could ever imagine to meet, even people whom I had beers during a master's. Thumbs up for the organization, and thanks a lot for the HiTSeq for giving us an opportunity to talk about this fun project actually, but you will see it's about serious matters. Yeah, so we are coming from Heidelberg, this beautiful city on the river Neckar, from the German Cancer Research Institute, and our division does cancer epigenomics, so we study all kinds of epigenomic marks in cancers, and primarily, of course, DNA methylation, because it allows to create such beautiful maps of tumor progression and create actually lineage trees, and these are highlights from our publications. And we also have a bioinformatic unit where we do sort of all kinds of computational method development and supporting our division. And as methylation is one of our primary data types, we use gold standard methods, and as you already heard, whole genome bisulfite sequencing is one of those. It's been published. The first whole genome methylomes were published in 2009, and actually these figures, which I collected, some of them are outdated already, show that it's still, after 10 years, rather niche application, but still remains important and very interesting for us. And it's actually, if you think of these deposited methylomes in the public databases, then you can extrapolate that many people kind of use the process data and do the analysis with it. It's important to note. And of course, single-cell approaches, as Jonas just mentioned, are coming up, and they essentially use, with some modifications, the same methods. Yeah. So very briefly, I mean, you saw this already. I'm very lucky. I can save time. So the principle, you have methylated cytosines with, after bisulfite and PCR, they get converted. Vice versa, they stay, and the unmethylated ones get converted. And actually, in our lab, and I think in many other labs, we have this standard whole genome bisulfite protocol. It's actually, in principle, requires a lot of input material, 0.5 microgram, and it's well established. And this is usually what people understand. And standard bisulfite. There are other protocols like segmentation-based whole genome bisulfite. We also kind of were co-developers of this method. It's a low input, allows to process like 30 nanogram or less, but the adapters are introduced via segmentation, and there's a gap repair step, which is filled with the genomic with the unmethylated cytosines, which are complementary to the genomic sequence. Yeah. So we decided to focus on this boring part of the workflow because many other people directly skip it over, and as you just saw, I mean, this part might be still underexplored. So we are talking about the step from the reads to these methylation calls, just like in the previous talk. So what are the steps? Actually, if you try to categorize everything that's been published and what people did so far, it's 4 essential steps behind trimming, and filtering after the alignment are things which are roughly the same as for genomic sequencing. Everybody knows you have to remove the adapters, quality trim the reads, and after the alignment, remove sort of these duplicates, the reads which start and end at the same position, probably originating from the same fragment. So there are peculiarities about the alignment. Again, 3-letter alignment, so reduced alphabet alignment, and some standard off-the-shelf aligner, and more sophisticated approaches. So FAME should have been here as well. And there are other things you should think of, like multi-mappers, repeats, and so on. The calling steps, so there are also possibilities. Some people do simple counting, some people do more sophisticated things like local realignments, and SNP calling simultaneously, and some do more advanced modeling. And there's, of course, this M-bias issue mentioned in the question. So the M-bias is linked to the fact that if you plot the methylation along the position in the read for all reads, yeah, so you roughly expect something like this. You would want to have no dependencies, so flat graph. The reality, and this is something from our data, looks rather something like this. So towards the end of the read, you often get decrease of methylation. This is like in segmentation data, this is directly related to these gap repair things. So if you have a piece of read filled up with the unmethylated cytosines, this will decrease your methylation level artificially. You have to take care of it. Yeah. So if you think how many tools are actually available for these steps, we're creating a database for our project. And only with these numbers, you arrive at like few pure combinatorics, thousands of workflows. Yeah, it's of course like totally overestimated because you can't combine everything with everything, but nevertheless. So it's many more than 5 or 6, yeah. So we sort of decided to take this in, totally unmanageable task because, yeah, if you expect, well, what's going on there, let's look into the workflows. You think it's very nicely ordered and structured, but in reality it looks rather like this. But still, it's a fun project, so we were not afraid, and we asked 2 actually major questions. Does it matter at all with which workflow you process this data? Are there any significant differences? So there was a spoiler already, perhaps. The second question was, is there any kind of specificity? So is there performance dependency on the type of protocol or biological nature of these samples? Yeah? And like more technical questions, when we go in for it, is, yeah, what is the way? What is the unbiased and reproducible way to do this with this type of data or other data types? And even the more important, what data can we use as the gold standard? So here's something which sort of motivated the whole project, because simultaneously when we were starting this, there was this marvelous study published by our old friend Christoph Bock. It's the biomarker assay study. And there they actually selected roughly 50 loci across the genome and performed a lot of locus-specific assays. And I have to say they're much more precise than the ones which were mentioned already, like arrays, genome-wide or whole genome bisulfite sequencing. So you have assays where you have 10,000 reads piling up at one position, and it's much more accurate. And so they had a number of samples, and since the division was involved, we had access to it. So what we did, we took actually 4 samples out of this very same set, so the characterized ones, and sequenced them with these 2 protocols I mentioned. Whole genome bisulfite and segmentation whole genome bisulfite. And in the whole genome bisulfite, we got like 1 billion reads per sample, and the segmentation, we got 300 to 400 million reads per sample. Okay, so this is what we consider to be one way to look into this problem having ground truth. Yeah? So our overall setup is that in the phase 1, we decided to sample this huge space very sparsely with the workflows of the people we worked with, we know, and yeah, it was a little bit biased by our network, but nevertheless. So in the phase 2, we plan to do a more exhaustive benchmark in controlled environments, so using computational resources available to us to run most of the things or most of the feasible workflows. And in the last phase, which is still a vision, we want to transform it to a sort of a long-lasting, endurant benchmarking where novel tools like FAIN2 or Bismarck 3 can be benchmarked against the data sets we're using, okay? So actually this is work in progress, and I give you sort of a sneak peek at the phase 1 results. So this is how we approached it. We had our data, we distributed it, and people ran. We had no control over computational resources. They did it the best way they could, actually, and returned us the methylation course. So which workflows did we cover? So here they are, and actually, you already from the previous discussion know that there are differences between aligners, and also methylation calling software is also versatile. So I think we have sort of what's important, we have workflows used in big consortia like IHAC, Blueprint, and Deep, and we have good sort of representation of wildcard aligners and three-letter aligners, so the reduced alphabet, extended alphabet, and different kind of methylation calling software, sort of counting with this M-bias determination, 2 modes for this Salan pipeline with the G-snap and so on. So this is perhaps not the most optimal sample, but something we had access to. So what came out? We collected all these variously formatted BAM files, BD files with methylation calls, invested a lot of time into reading them in and solving all the problems. So what did we get? So basic stats, how many CPGs do we receive from the very same data? So these are our workflows, and so the x-axis millions of CPGs for both protocols. And you see, wow, already here you have like a span of more than 10 million CPGs, like between the GMBS workflow and the BAT, which is actually Ziggy-Mail in the previous talk, is the aligner behind it. Yeah. So as you see, you don't want to lose 10 million of CPGs out of the blue, so there might have to be reasons behind it. Yeah. So next, what is actually the coverage, the depth of coverage at those CPGs? And yeah, these are the values. Actually, this surprised us a lot, but that you probably have noticed it's the only workflow which didn't do this removal of duplicates, although it's still puzzling because it's way more than one would expect even under these conditions. But yeah, you see there's still, again, a span, a dozen in counts that you lose the depth of coverage, and a bit more variation in the less dense data. Yeah? So already at the level of the most basic quantification, there are differences. Okay, so the first actual methylation measurement we can just do a mean across the whole genome, yeah? Just average everything and get some. It's actually a very important biologically driven statistics, actually. Do you know that cancer cells usually have lower methylation genome-wide and so on? In this case, not that much, but I mean, this value can't be totally random. And as you see, like globally, these workflows get it roughly the same, but if you zoom in, there is definitely a variation of like a few %, and it's systematic, as you can see. So the order of samples stays the same. It's just that it overall shifts, which indicates probably there is a bias behind it. And we think it has something to do with this M bias, which is quite a complicated issue, which I explained. And one more thing is that here we can already start using these data from the benchmarking study of biomarkers, because they also perform the global estimation. Yeah? And then you see that, that most of the workflows underestimate. If you calculate the deviation, you can sort of rank the workflows for the first time, and you arrive at something which is different from the ranking you would derive from basic statistics, but nevertheless. So this was our second discipline. And then actually the core analysis for which we were aiming is the analysis of these loci. And here is how they look. The authors of the benchmarking study aggregated methylation per each region. So it's a mean in this region, 48 loci we have only. And what they proposed, because they had, of course, a spread of values from different assays. They derive the consensus corridor, which is the smallest region, which covers 3 measurements from 3 different assays. And they assume that here actually the ground truth methylation value resides. And what we did, we calculated the deviation from this consensus corridor. So if a workflow, the whole genome bisulfite workflow is in the corridor, it's 0, and then any absolute deviation is counted. So this is what comes out, and ranked by sort of mean across 4 samples. So it's all in a summary, and as you can see here, they perform roughly the same, but still, yeah, you would rather use something on top than at the bottom. So this is whole genome bisulfite sequencing, very dense data, 50x coverage almost. So surprising thing, if you switch to a different protocol, which is like less quality data, less dense data, the ranking almost inverts, or at least the workflow which was the last in the previous case becomes the first one. I mean, we are still looking for an explanation, but this is what you get if you're kind of looking at things unbiased. And behind it are, of course, all kinds of things happening. I just give you a few examples. So there are regions where things are pretty consistent, so different colors now mark different workflows, but it's not important. So first one is pretty easy, probably. The second one is more complicated, but most of the workflows get it right. And there are, of course, regions where things go quite differently. So we are still looking into this. And yeah, this is the way we want to continue adding new workflows and seeing how they behave. And of course, you can say this is limited to the used regions, and you will be most probably right. And we are still thinking how to extend this to more CPGs. And yeah, this is quite difficult. I can show you in the backup slides why Illumina might not be the best way to do it. But if you just look, again, abstract from the gold standard and look at genome-wide differences, yeah, this is sort of a discordance score, which is basically an absolute difference, again, between the workflows plotted. That you can see that if you look carefully, you can see that there are clusters forming, and in many cases, so these are related workflows which come into it. So yeah, so these were using the very same aligner, for example, and the same here, and here is the very same workflow with different parameters. So obviously there is a signal in this data, so the workflows do matter, and that's why otherwise everything would be through each other, naively thinking. So there is something to extract there. And yeah, just a bit of vision. So you can, of course, use each of these categories to rank the workflows, like just we do after every Olympics, who got more medals here and there, and derive a final rank. And it will be different, as we saw, between different data from different protocols. So like if you extrapolate a bit and think of this holistically, we want to arrive at something like a database and an interface which would allow you to, in a personalized manner, select the workflow which performs the best for your particular case, yeah, and get a list of priorities. So in summary, yeah, the existing workflows show differences. There is variable coverage, depth, and to some extent accuracy. Performance is specific, at least from what we saw so far, to a protocol. Yeah, these locus-specific measurements, they are valuable, but it's still limited resource, so we would need some bigger ground truth to be more general. And yeah, it's actually these meta-analyses or large-scale integrative studies which use data from here, from there, from wherever as calls should take caution based on the talk of Jonas and what we see, because you can get funny differences just because of different processing. So if you do such analysis, better try to get the reads, but we know it's becoming increasingly difficult for human samples, for example. So this is a problem. So I'd like to thank our team. So we are this computational unit inside the division. Reka, who spent a lot of time on this, and the interns, and of course, all our collaborator partners. The list is probably longer. Sorry if I didn't include everyone. Yeah, our division head and Dieter, who did all this sequencing. We actually managed to get funding for this fun project from BMBF. Thanks a lot from German ministry. And one more important thing, so Reka has a birthday today, so I really appreciate your effort, and we'll celebrate later. And the last thing, we are a very vibrant community in Heidelberg. Lots of opportunities for great bioinformatic computational biology research. So we are looking for people. Please contact me if you think of working on something like this, and yeah, let me know. Thanks a lot.
Thank you. Very interesting. Are there questions? Please come to the microphone.
Hi, Marcel.
Hi, Pablo. So this was really interesting that you were showing then when you switch the protocol to segmentation that it really inverts. So do you think that in this segmentation WGBS there's much more noise, and so that's why the previously method seemed to, according to your metric, has the fewest mapping positions, but probably does much more filtering, quality-based kind of filtering, then it's turning out better? So probably
that's
even was tuned.
one explanation. Yeah, so I have to admit our design was a bit confounded. You must have noticed it. I had a typo in this diagram. That's why it was maybe a bit confusing. So we used two protocols, but also two different sequencers. Yeah, so one right should have been segmentation, and for segmentation we use the old established sequencer, and there we have a HiTSeq. I mean, it's just those are just facts. I can't interpret it right away for you, but this might have contributed as well. So that they optimized their workflows for HiTSeq 2000 data, and Xten is slightly different. It's more complicated actually. We have more reads, but the error rate is much higher in Xten. So this was the only thing reasonable in terms of cost and covering all opportunities. But we still have to maybe sit together and look at it in detail and derive some interpretations, because also maybe with GMBS people. Yeah.
Are the reads of the same length in both technologies, what you sequence?
Xten reads 150, I think those are 125. Yeah, those are shorter reads.
Okay, there's another question.
Thank you for the talk. Do you think it's realistic to automatize the choice of the combination? And do you have any ideas how to approach that?
Yeah, so we were contemplating about it already. We have some heuristic which might help to sort of find ways, and not everything will be compatible with everything for sure. So there we will have to do cuts, but at least like trimmers should be kind of compatible with most of the aligners, and but this will be quite a task. I can't promise we'll manage, but we'll do our best. We now promised it to the funder, so we'll try to make the best of it.
Thank you.
Follow-up question to that. So I guess you could maybe run more combinations. So and I guess you could probably also, I don't know, fit a linear model to different components and figure out maybe interaction terms and so on. So I mean, if you would generate this data, it would be a great resource for doing.
Yeah, that's what our aim is, just to provide a resource for the community, and for ourselves, you want to know how to continue with those data. And single cell, if we could get single cell data from, again, those samples would be great. We are trying to approach that, but.
Okay, there's one last question.
Hi, are you using any kind of a workflow language for the workflow, and do you plan to make it public so other people can use the workflow?
Yeah, it's kind of almost a prepared question, but I don't know this person. Yeah, I have to say we are increasingly using common workflow language for all our work, and we highly recommend. Actually, I didn't plan to advertise it, but we actually have a poster on that, and Ignite talk, so on our tools for CWL. So we think to do this automated step using CWL wrapping, and this is probably the answer to the question.
That's great. So on this cheerful note, let's thank the speakers of all the speakers. So the session will continue at 4:40. However, we will have an industry talk, a sponsor talk, at 4:25. So please come back. AWS will talk about using AWS for genomics in the cloud.